Laboratory of Microbiology in Delft.
Live streaming is on.
Okay. The history of the microscopy with Antonie van Leeuwenhoek, and we are very glad that she's going to present her vision on how the historical microbiology can still contribute in this 21st century microbiome research era. Leslie, the floor is yours.
Humanity has been playing with microorganisms for a long time, whether they knew it or not. This is actually considered to be the first piece of written evidence there is to make notes that what people were producing. And if you look there, you can see the Mesopotamian symbol for beer. And that was about 4,500 years ago. After about 4,000 years, we come to Delft and we come to a linen merchant who was rapidly becoming obsessed with his homemade microscopes, which you can see here. I've included a flower simply to give you an impression of the size of these things. They're very simple. You can see that there's a lens at the top and then there's a needle for samples and focusing screw, but not an awful lot. He discovered using these tiny little microscopes, bacteria, protozoa. I've included yeast on the image because, of course, this is a biotechnology department, and everybody plays with yeasts these days. After about 150 years, people were finally beginning to agree that maybe microorganisms had something to do with disease. It took an awful lot of time and a lot of trolling. That's another story. People started looking for industrial applications and also to see what was going on in the environment. And there was in Wageningen at one of the agricultural schools, there was a botany teacher whose life was about to change. Martinus Willem Beijerinck was recruited to start the very first industrial microscopy and microbiology lab certainly in this country, and I think in most of Europe. He was recruited by Jan van Marken of the Delft Yeast and Spirits Factory, and his wife, although in those days women were not allowed to own things like factories, so she only got to play in the background. Beijerinck, unfortunately, was a bit of a scientific butterfly. If anything went past his view, he would be looking into it. I don't expect everybody to read and remember everything on this slide, but you might want to take note of the ratio of red text to black text, because the red text is what he was supposed to be doing, and the black text is the rest. And his lab journals are almost impossible unless you've got a lot of time, because he'll label one book bacteria, and then on page 5 he'll go on to something else, and then he's back to yeasts on page 9. And he's sometimes even on the same page. And I mean, his unyeast work, enzymes, root nodules, all sorts of things, even sunsets. He had a paper in Nature on whether Krakatoa volcano and its dust was making the glorious sunsets they were having. Eventually, he was getting very depressed because he wasn't having that much success in industry. And Van Marken talked him into becoming the first professor of microbiology at the Delft Polytechnic. This is the Delft Polytechnic in its later incarnation, of course. And this is the first laboratory. It's not quite as embedded in woodland, it's sitting on the edge of the canal. Beijerinck achieved a lot, but they only gave me 30 minutes. So I've been picking out highlights from him and his successors. I think one of the most important things he did is the enrichment culture. We're still using them today. I certainly use them during my own promotsi, and I'm pretty willing to bet that many of you listening to me now are also doing it. I'll come back to it in a second. He was also the person who isolated and did the initial characterization on a lot of key bacteria that we still play with today. And the one that everybody remembers, and he would have said it was one of his least achievements, he demonstrated that viruses were self-replicating, so they must be some sort of biological life. The picture I put there because it shows both sides of the sulfur cycle, and that is one of the ways I started out. You can see these little red spots all over the place. That is actually molecular sulfur and appears in quite a few sulfur oxidizing bacteria at some stage of their metabolism. It's quite an impressive picture though, because Henrietta Beyerink showed them as pink. And I only know one way for that sort of sulfur to show up as pink under a microscope, which is to use phase contrast. And that was still 50 years in the future. How did she know to do it? Anyway, enrichment cultures, very simply put, you decide what you want your bug to do. For example, we were asked in the 20th century to find a culture that would handle photocopy toner. And so you take your liquid, you add a little bit of whatever the strange compound is that you want and other things. You choose the culture conditions to suit the organism. Would it do better at 5 degrees? Would it do better at 75 degrees? And then you choose your inoculum from somewhere that might favor that sort of bacteria. If in doubt, I always went to and took a soil sample from over near Europort. There's everything in that soil. And doing this sort of thing, Beyerink came up with all sorts of things: sulfate reducers, sulfur oxidizers, all sorts of things. Enrichment cultures are used all over the world today. Every wastewater treatment place will have them. But it's surprising how rarely it's mentioned that it was Beyerink that came up with the idea. And then there's the viruses, of course. 2 other people who had been trying to work out what these unattractive blobs on leaves are. This is from his thesis. And they decided it must be chemical because they couldn't see anything, they couldn't find anything, but they asked him to confirm it. And what he did was to take infected leaves, grind them all up and put the liquid through these porcelain filters. And the smallest of these is good enough to stop all bacteria. He'd take the filtrate, put it on healthy leaves, and they would get sick. And by the time he's done that 6 or 7 times, I know all you 21st century scientists are saying he would have diluted it out if it was a chemical, but it didn't, it got stronger, so it must be self-replicating. However, oh yeah, nearly forgot it. This is probably one of the most famous lab journal pages in the world. I've given so many people permission to use the image, I can't remember how many now. This is one of 2 pages from his lab journal, which is all that there is, and I've gone through all his lab journals, talking about tobacco sickness disease and talking about the rest of it, because he couldn't see anything. He couldn't do anything except make plants sick. And being Beyerink, he got bored and went on to do something where he could see things and do things. Gerrit van Ittersen was one of his PhD students, and his big obsession was mathematics. This is one of the men who invented biomathematics, and I have been known to curse him on occasion. The image shows what happens when you slice through flower buds, and you get all these different patterns, and every flower bud has its own pattern. And you can analyze these mathematically, apparently, to work out how they're all packed into their different buds. And a smaller achievement of his, like Byrink, of course, I'm only picking out highlights. He started the Delft Botanic Garden, which is quite important because it's the only it's the only one I know of that is actually a botanic garden for applied work. They only deal with stuff that has a use. And it's just over there. Van Eterson's doctor's thesis, he finished about 103 years ago, and we're still getting requests for copies of it. Although please don't ask any more, you can go to the library website and download it, and all the others. The big thing was phyllotaxis, the way things grow that make spirals. And they became known as Fibonacci phyllotaxis. He strongly believed that professors should be teaching and not doing much research. And so the rest of his publications on this subject had to wait for him to retire. But he came up with another 4 or 5 publications about it after that. I put a sunflower in because the weather's so miserable instead of a plant. And then the third of them, it's getting a bit incestuous because Cliver was a PhD student of Van Eterson's, and he became the second professor of microbiology after Byrink retired. Again, lots of achievements. Again, these are the star turns. Unity in diversity, which when he wrote a book, he called Unity in Biochemistry. And they showed that the function of an enzyme or a biochemical pathway actually defined the structure of that enzyme or pathway. People used to think that every single species had its own different set of enzymes. And he showed that you could take an enzyme from an insect and put it in an elephant. Well, he didn't put anything in elephants, but in mice. And they operate. And it's true, they're still doing it to this day. But they were the first people to realize this. The laboratory actually sent out Christmas cards that year with an insect and an elephant. And the slogan from the insect to the elephant, it's all the same. Then what I think is also an important one, which he never seems to get credit for, is submerged cultures. It's for heterogeneous cultures. They used to grow fungi and therefore fungal products in bottles. And you would get them just like this. They lie on their side in the incubators. So you've got sometimes a whole temperature gradient of maybe 1 degree between the edge and the center. The darker bits, it's where the fungi are sporing. The whiter bits are new hyphae. So, of course, no 2 bottles were ever exactly the same. And it's not easy to do industrial things like penicillin production using this sort of system. And so Cliver came up with what is now known as the Cliver Flask. You take an Erlenmeyer flask, you turn it upside down, put a glass filter in the bottom to break up the airflow, put an access port in the top to let you pour things in. And these things are being tested, and when they're set up correctly, the KLA is just as good as with a 21st century fermenter. The guy from Applicon admitted it. We've done this with 5-litre Erlenmeyers and put extra ports into what would be the base of the flask to take electrodes and things so that you could actually run one of these off a couple of aquarium pumps. And I know also that way back, there was a group desulfurizing coal biologically in microbiology, and they were using them with coal with pyrites in it and growing them with sulfur oxidizing bacteria to keep everything going. It worked fine. Some people don't like it when things are that simple, though. This poster I've included simply because I think it shows a light on Cliver's character. Remember that this was a time when they treated professors very formally. You know, if a professor walked into the room, everybody stood up. And so it's refreshing to see that his staff and students were reluctant. Oops, wrong button, excuse me a second. Back. Yeah. His staff and students felt safe enough to give him a gift token for a canary. He was a chain smoker and using cigars when he could. They also teased him about the fact that he set fire to his bed once, smoking in bed. And so it went on, his books there, the reason for the block of yeast and the soap. In World War II, when the hunger winter hit this area, the Hisporocardas, as it was then, were feeding their staff on soup made from compressed yeast. And as one of their advisors, Cliver could go around there and get his bowl of soup every day. I don't think, I met, I don't know if you still use Schlegel's book on microbial physiology. Well, last time I saw Schlegel, he said, you know, you've got to remember one thing about Cliver. When a little German student was trying to get out of East Germany, he met a Dutchman who had every reason to hate him, and he didn't. He helped me to get going. This was the top microbiologist in Germany at that time. So back to historical microbiology. Historical is often added these days to a subject to show that it's been investigated practically as well as in books. Nothing wrong with books. But you see presenters dressing up in period costumes and going out to show how to plough a field with a pair of horses, or to run a kitchen with Victorian cherry molds and things like that. We don't go that far in historical microbiology. We make them keep their lab coats on. And it's been, oh, it saved us a couple of times. The first year practicum used to include a trip down to the city to take canal water samples. And they could then learn how to identify them. And then we had a seriously hot summer, hot enough that the canal levels all dropped. And several students managed to find Clostridium botulinum. Now, according to the biological safety laws, you're not allowed to let teenage students play with level 2 pathogens. And so we had to rewrite chunks of the practical. And what I did was to pick up 2 historical experiments. We dumped the canal water and gave the van Leeuwenhoek's work with pepper water, which was in 1669, and Beyerink's use of bioluminescent bacteria as oxygen probes in 1889. The van Leeuwenhoek one is very simple because he was a bit economical with his experimental detail. And so we gave each student a copy of the first letter. This point about this experiment is that this is the first experiment that turned up bacteria in his microscopes. So we gave them a copy of what he did. They're told they're doing re-research, and we asked them to work out how he got the results he got with peppercorns. He'd actually been soaking the peppercorns in water to try and get them soft enough to cut, but he couldn't. He just got cloudy water. So, of course, that went into his microscope. And that the students are allowed to select items from a list. You can't just tell them they can do what they like because the media kitchen will murder you. So they got a choice of water types from snow to tap. And just to see who thought of adding controls to their experiments, we added sterile demineralized water just to see who used it. They were given simple analytical tools like pH paper and those dipsticks you can get if you want to measure ammonia or nitrite in the field, that sort of thing. They could choose agar plates just to see who'd been listening when we talked about enrichment. So they could have rich peptone plates, or they could have all sorts of other mixtures, right down to mineral medium that's only got pepper as the total energy and carbon source. Two managed to turn up bright purple colonies on that. We've never seen them again. I've never worked out how they did it. But they could choose the pepper. Van Leeuwenhoek obviously only had black and white peppercorns, but they could, some of them want to use the modern red ones and green ones, and they could choose whether they wanted to use them whole or ground. Using a Van Leeuwenhoek microscope is an acquired taste. And also, there's no way we could afford to buy 16 Van Leeuwenhoek models, not even the copies. They cost €250 each. So they were given the monocular compound microscopes that Cliver used to use with his practicants. And everybody came up with results. And not only, they came up with such good results that we actually put a poster up at a microbiology meeting signed by me and 700 and something students with all their results on it. But the thing that impressed me most, they were supposed to be with me for 2 days a week for 5 weeks. They did some of their studies in Leiden and some in Delft. And it seemed that almost every time I walked past the practical room, there were students in there visiting their bacteria to see what they were doing. They didn't have to do it, but they all came in at one time or another. They even looked guilty if I caught them. Then you come to the bioluminescent ones. This is the sort of bioluminescence that you get in the sea. Virtually every sea fish has it on its skin. And I would trot down to the fish market, because that's where Bioink used to get his fish as well, and buy up half a dozen sea fish, and the students would be challenged to isolate bacteria that can make light. They worked at 15 degrees or lower because some low-level pathogens like Vibrio cholera can make a little bit of light at 30 degrees, but not below. And they're told you can't get pure cultures in 5 weeks, but this is practicum land. It's not the real world. So whatever you get, you can pretend it's pure cultures, because we wanted them to try out very simple traditional taxonomy, so they have an idea of it before they get on to more complicated things. They get DNA analysis in other practica. They grow them in shake flasks, and you get beautiful light-making cultures, and then they're transferred to glass measuring cylinders, narrow ones, and left to go dark, because as the oxygen runs out, they stop making light. Then you add 2 drops of hydrogen peroxide, and you get this band of light that goes down the measuring cylinder. And then eventually the whole culture lights up. Then you take a second measuring cylinder and let it go dark. And then you add a piece of seaweed, which has been sitting in the sunshine for an hour or 2. And that's what you get. You can see the seaweed with the lights on, turn the lights out, and for about 15 minutes, you can get light exactly where the seaweed is, because the photosynthesis can continue that long without light. Great fun. And again, if you want to get teenagers excited about things, give them flashing lights. They love it. So then I spent about 35 years as curator of the Delft School of Microbiology archives as well. That was a volunteer job. And all of that time I've been getting, could you please look up how they did this, who did that, or whatever. And this one I loved, because this guy wrote from California, and he said that he was told that the algae he was working with had come from Bioink, but he couldn't prove it. And yeah, reviewers, they're always difficult, and they were being very difficult about this. So we spent a couple of days in the correspondence archive, and we could show that the algae had actually come from Delft, but they'd come from Cliver, which is a puzzle. He hadn't worked with them. Further search took us to Professor Pringsheim, and he was one of the big European algae specialists. And when he and his wife had to escape from Prague in 1938, in order to be able to bring his culture collection with him, they dumped everything they owned except her jewelry. And they arrived in Delft with a case full of algae and not much else, stayed a few days, and then went on to Cambridge, where he spent the rest of his career. But while he was in Delft, he had handed this alga to Cliver. And so, yes, it was one of Beyerink's cultures, but it had a certain circuitous travel to actually get to America. Visiting researchers around the end of the 20th century had a big ambition. They wanted to start their country's first national culture collection, but they had no liquid nitrogen. They had about 4 hours reliable electricity a day. So what on earth were they going to do? They came on a visit, and they knew that Beyerink had been preserving his bugs before they had reliable electricity. So they wanted to know what he'd done. And I knew because I'd been warned they were coming. But they used to dry their cultures onto sand or onto starch or even onto dried milk, and then seal them in glass tubes. The reason I get showing you this one, even though this is Pseudomonas pyogenes on starch powder done in 1926. I don't know if this one's viable. I've sent them all off to the Dutch Culture Collection now. But it was showing signs of something very typical of our collection. It was packed in a cigar box. And one thing we got used to doing, anything that was reasonably small in our whole collection, whether it was cultures or chemicals or whatever, was packed in cigarette and cigar boxes. And this one was a nice color and it's got the date on it. So I thought I'd show you that one. But those Photobacterium splendidum cultures you were looking at came out of a similar tube that we happened to have in Delft about 15 years ago. Then I had a personal problem. My bugs would not stop denitrifying. Didn't matter what the dissolved oxygen concentration was. Turned out this was a phenomenon called aerobic denitrification. And once every 10 years or so, somebody's put their hand up and said, I've got aerobic denitrification. And everybody else has said, oh, no, you haven't, and dumped the idea. But thanks to the university in Amsterdam and in Aarhus, we managed to take very modern equipment to it, and we could show that we did, but we had to prove that there were no anaerobic microcytes in our thing, because somebody actually stood up at a conference. I wasn't speaking, my boss was, and having sat through the whole of his lecture, said, don't you know this is all impossible? But hey, so we set up a culture of Photobacter splendidans in a very rich medium with not a lot of headspace because I wanted to give it the highest BOD I could manage. And looking at it with the lights on, they looked identical. You couldn't see any difference between them. You know, I challenged people to tell me which one was which until you put the lights out. And there you can see one very brightly glowing one because it's aerobic throughout. The other one isn't even aerobic at the bottom of the pot. And you've got these blotches of darkness all the way up the culture. And that one was stirring at half the speed of the other one. So you do get anaerobic microcytes in these cultures. You've just got to watch your stirring speed and your aeration speed. So historical microbiology can be, it's fun. It's great fun. It can be useful. It's a good reservoir of experience. And I've also found that at big science fairs and things, the public are not so intimidated if you've got historical stuff on your stall as well as the modern stuff. They actually stop and ask questions. But if you go back to my second slide and rotate that Etruscan tablet and look carefully at the symbol for beer, you've got yourself a Cliver Flask. And it seems to me that Delft School of Microbiology actually goes back before van Leeuwenhoek. It goes back 4,500 years. And so I'm not saying historical microbiology is a substitute for anything. Obviously, you need the new wonder tools, and obviously nothing's a substitute. Although we did one year have a load of students make a protest because they said the microbiology prac was just chemistry with bugs making life difficult, which is when we added the photobacter experiments. I think you got to quote Professor Beyering, gelukkig zij die nu beginnen. Happy are those who are starting now. Thank you.
Thank you very much, Leslie, for your wonderful story. Are there people online or in the room who have some questions or discussion points or want further enlightenment? I can't see if there are.
My TU Delft email works if you want me.
Okay. Yeah, David.
How was it proven that the culture came from Delft, this algal culture? So did they use sequencing at that time? Or what could they use to prove the culture came?
I'm not going to embarrass him. He was supposed to come to Delft, but managed to leave his laptop at Los Angeles airport and had to go back for it. So I still haven't met him, but I'm not going to. It's in one of the universities along the Californian coast is the furthest I'll go.
It's a contemporary. So
yeah, since we moved into the Science Centre, so in the last 5 years.
Okay, I thought it was
oh no,
years ago that.
The earliest one was my own one with the BODs and the chemicals and the photobacters.
Thank you.
No, you don't embarrass people like that. I'll tell you about the lot that tried to isolate sulfur bacteria and nitrates and did all their analysis and isolation using peptone agar and peptone water and was surprised when nothing grew. And all there was in the soil they were looking at was sand and pyrite. So.
Any other persons want to make a comment? Just open your microphone if you're online and start speaking. That's okay. Sergio.
Hello.
Hello.
And I was commenting with them because before we came to Delft, like already some years ago, we didn't know anything rather than, for example, the microscope. But there are many things that has happened here in Delft, and we only knew once we came here. Mm-hmm. So why do you think that is not as well known outside just
I think people tend to only read the literature relating exactly to their own subject these days because there's so much of it. If you search for me and microbiology in Delft, you'll find all sorts of papers and reviews. I've got about 7 or 8 about van Leeuwenhoek alone out. That's Fem's Microbiology Letters. We even had a big exhibition in the new old church in Delft where we pointed out that Delft is actually the birthplace of microbiology. This is home. Some people actually turn up in my office saying it's so good to finally come home. And they're just microbiologists coming in from 1 or 2 countries who take their history more seriously than the rest of us. But it's good fun.
Thank you.
Thank you. It's good. Enough self-promotion for the time being. And maybe to put things in perspective, I had a few years, well, quite a while ago, visiting some famous American professors saw the Van Leeuwenhoek-laan and taking pictures in front of a house. The women came out and said, "Why are you putting my house on this picture?" She didn't know who Van Leeuwenhoek was, and probably that's also what Delft in generally does, forgetting their history. But thank you, Leslie, and anyone who's interested to chat further, I'm quite sure you can find her email address online. You can find a lot of information online, and she's more than happy to interact with you, to send her emails or what else you like, if you like to discuss history of microbiology.
I'm usually in the Science Centre on 1 day a week at the moment. So we've just done a rescue on a cellar full of stuff that a professor had taken with her when she retired that she shouldn't have done. And her executor rang up and said, "Please come. I don't know what to do with all this lot." So I'm quite happily playing with the 19th century microscopes at the moment.
Okay, thank you again. Okay, we will now move to session 5, and the easiest way